Hello, dear students. This lecture is about population genetics, and we will be discussing some basic terms and problems related to population genetics. So, the Hardy-Weinberg law, which is from population genetics, this is a law of genetic equilibrium which provides a mathematical model for the study of evolutionary changes in allelic frequency within a population. Hardy and Weinberg in 1908 independently proposed that frequency of alleles and genotypes in a population will remain constant from generation to generation if the population is stable and in genetic equilibrium. So, they proposed five conditions which are required in order for a population to remain at Hardy-Weinberg equilibrium. And these five conditions are that the population should be a large population and it should be a breeding population. So, a large breeding population is first parameter. Second is that there should be random mating between the individuals of that population. Third is no change in allelic frequency will happen due to mutation. So in that population there will be no mutation so that the Hardy-Weinberg equilibrium is maintained. Then fourth is no migration or immigration, and fifth is no natural selection. So no immigration or immigration means that no one should go from that population and no one should enter in that population, and no natural selection means that the individuals will be all will be surviving in that population. So these are the five conditions. So the first parameter, which is a large breeding population, it helps to ensure that chance alone does not disrupt genetic equilibrium. What does that mean? It means that if a population is small, then only a few copies of a gene or a certain allele may exist. If for some chance or change a reason the organisms with that allele do not reproduce successfully, then the allelic frequency will change. And this random non selective change is what happens in genetic drift or bottleneck effect. It means that if the population is large, then even a small change will not matter for any allelic frequency or gene frequency. But if the population is small or the sample size is small, then it will affect the overall genotypic and allelic frequency. Then the second parameter is random mating. In a population which is at equilibrium, mating must be random. In assortative mating, individuals tend to choose mates similar to themselves. For example, the larger blister beetles tend to choose mates of larger size, and smaller blister beetles tend to choose smaller mates. Though this does not alter allelic frequency, it results in fewer heterozygous individuals than you would expect in a population where mating is random. It means that in a random mating population, the number of heterozygous individuals are more compared to the homozygous individuals. That is why a random mating population is desirable with a large breeding population. Third parameter is that there should be no mutation. When there is mutation, there will be a change in allelic frequency. So, the third parameter of Hardy-Weinberg's law is that there should be no mutation in the population so that there is no change in allelic frequency. For a population to be at Hardy-Weinberg equilibrium, there can be no change in allelic frequency due to mutation. Any mutation in a particular gene would change the balance of alleles in gene pool, and mutations may remain hidden in larger populations for a number of generations but may show more quickly in small population. It means that even when mutations happen, if the population is small, it will have a large effect on small population, and if the population is large, then the mutation, though it may be occurring, it because of the large population, it will not show. Then the fourth parameter is no immigration or emigration. For allelic frequency to remain constant in a population at equilibrium, no new alleles can come into population and no alleles can be lost. Both immigration and emigration can alter allelic frequency. So, a fourth parameter says that for a particular population, no one should come and no one should go from that population, so that the Hardy-Weinberg genetic equilibrium is maintained. Then the last parameter is no natural selection. It means that in a population at Hardy-Weinberg equilibrium, no alleles are selected over other alleles. If selection occurs, those alleles that are selected will become more common. For example, if resistance to a particular herbicide allows weeds to live in an environment that has been sprayed with that herbicide, the allele for resistance may become more frequent in that population. It means that fifth parameter says that for any population in genetic equilibrium according to Hardy-Weinberg, there should be no natural selection. It should be as natural as it can be. So you can refer any book of genetics which describes population genetics, and it is nicely described in Pearce, so you can refer Pearce for this particular topic. Now, as we have just discussed, what is Hardy-Weinberg's law? So Hardy-Weinberg's law is based primarily on 5 parameters, and these 5 parameters you can see in this book, that these 5 parameters are your randomly mating population. If a population is large, this is the first parameter, randomly mating, then not affected by mutation, migration, or natural selection. Then there are 2 predictions. Prediction 1 is that the allelic frequency of the population do not change, and prediction 2 is that the genotypic frequencies stabilize and will not change after one generation in the proportion p square, which represents the frequency of homozygous dominant, to pq, which represents the frequency of heterozygous, and q square, which represents the frequency of homozygous recessive population. So where p equals to the frequency of allele A, which represents dominant allele, and q represents the frequency of allele small a, which is recessive allele. So there are 2 equations for Hardy-Weinberg equilibrium, and these equations are followed in a population which has these 5 parameters. So there are 2 terms. What is genotypic frequency and what is gene frequency? So how to calculate genotypic frequency? This is the formula when the number of individuals are given. So according to this equation, the genotypic frequency of homozygous dominant will be number of homozygous dominant individuals upon total number of individuals. Second equation is that frequency of heterozygous individuals, it is number of heterozygous individuals upon total number of individuals, and the third one is for frequency of homozygous recessive individuals. So this will be number of homozygous recessive individuals upon total number of individuals. And when we add all these, the genotypic frequency will always be equal to 1. So in simple terms, this is p square plus q square plus 2pq, where p square represents homozygous dominant individuals, q square represents homozygous recessive individuals, and 2pq represents heterozygous individuals. Now, how to calculate allelic frequency from the number of individuals? So, here this is the second equation where we find that frequency of an allele is calculated like this: number of copies of the allele upon number of copies of all alleles of at that locus. So you can see here in 25.3 equation, the frequency of dominant allele, which is represented by P, is equal to 2n plus n upon 2n. So 2n homozygous dominant, it means that if we are calculating the frequency of dominant allele from the number of individuals, so 2n represents 2 into n, it means 2 alleles of homozygous nature, plus n represents 1 allele, dominant allele, and 1 recessive allele. Similarly for Q, that is your frequency of recessive allele, the same equation applies. So where n represents the number of individuals of different types: homozygous dominant, homozygous recessive, and heterozygous, and capital N represents the total number of individuals. In denominator, it is written as 2n because each diploid organism has 2 alleles. So allelic frequency, when we calculate from the genotypic frequency, this is the next equation which you have to remember. This is 25.4 from Peirce. So this is when we calculate the frequency of dominant allele from genotypic frequency. This is frequency of homozygous dominant plus half the frequency of heterozygous individual. Similarly, the frequency of recessive allele we calculate by frequency of homozygous recessive plus half the frequency of heterozygous individual. Why half the individual? Because the heterozygous individual has only one allele, each of dominant and each of recessive. So how to do some problems? We will see how to solve the problems of genotypic frequency and allelic frequency related to Hardy-Weinberg's law. So this equation we have just discussed, how to calculate allelic frequency from genotypic frequency. And say this is a work problem which is given from Peirce. So this is the question which says that the human Mn blood group is so and so, and this is the data which they have given with phenotype, genotype, and number, and they are saying calculate the genotypic and allelic frequencies at the Mn locus for the Qajarala population. So this is the problem from Peirce, and this is how they have solved from the equation we have just discussed. For genotypic frequency, they are doing this by putting this formula: number of individuals with the genotype upon total number of individual in the sample, and then we will calculate like this: 182 by 398 point, so it comes out to be 0.457, and so on. Then for frequency of allele, they have again this put formula, the second formula which we discussed, and then we have calculated the frequency of dominant and recessive allele by the third formula. So you can refer for this particular problem the book of Peirce, and we will be discussing some more problems in the next part 2 of this lecture. So thank you, students.